Okay, well, welcome to the last session of the day. And when I was first asked to present, I came up with this beautiful, grandiose title here that really no one could possibly address in 30 minutes. And I've decided that there was a niche that I think fit in well with the other speakers, and that is food-based cancer prevention strategies. And I think it's very appropriate given the number of folks here from industry that actually focus on foods. So we're going to talk a little bit about this niche. There's no conflicts of interest. You will hear many opinions, and I'm very grateful to all the organizations that have supported us over the years. Now, so there's sort of a conceptual framework that all of us working in the field use to organize our thoughts about diet, nutrition, and cancer. And today, you've heard beautifully and elegantly some of the issues related to dietary patterns and cancer. And this is really, really key, I think, for the future of public health recommendations for impactful cancer prevention strategies. And you heard from Dr. Hursting quite a bit about the energy balance issue. And this involves the macronutrients, the intake, unique aspects of metabolism, and the exercise or physical activity component. Now, in this realm of diet and nutrition, so many of us that trained over recent decades focused on reductionism. Okay, if there was an epi study that suggested a certain food was related to cancer, we needed to know what chemical in the food was responsible. And so much of our research efforts and funding priorities continue to support this reductionist thinking process. And much of the epidemiology now, as we develop new tools and new approaches and statistical testing to look at dietary patterns, I think it's very clear to me that for a public health approach, we want to focus more on a more holistic approach to the dietary prevention. Now, part of me lives in the clinics, and in taking care of patients or those who are at risk, whether that's due to exposures or genetics, I think there's a very important role for perhaps foods and chemopreventive agents, whether they be nutrients or natural compounds, all supported by, I hope, evidence-based research. Now, if we're going to work in this realm of foods and functional foods, there are some key assumptions and principles that we need to think about. And the first one that I have the most struggle with my fellow scientists in cancer particularly, is that when you're working with foods, you have to get comfortable with this black box, meaning the food is not one or two chemicals or even 10 or a dozen. They can be hundreds or thousands. And we have to get comfortable with that fact that we're not dealing with a single chemical entity. And there are a couple principles then that guide what we do. One is that there are multiple bioactives with diverse mechanisms of action that may have multiple targets and thus may be more effective than any one of those components alone. And secondly, multiple bioactives at a modest dose with non-overlapping toxicity provide a margin of safety. Now, these principles have worked very well in pharmacology. This is how we cure certain cancers. Look at childhood leukemia. It wasn't cured by a single drug. It was cured by multiple agents following these principles. And I think this is very appropriate to integrate into our thoughts about foods and cancer. Dr. Hursting reviewed the Hanahan and Weinberg essential characteristics of cancer. And when you understand that cancer has multiple different defects that all provide unique biological targets, you can appreciate how multiple components or phytochemicals within a food may also work by integrating across this diverse spectrum of biological targets in order to give us a more effective outcome. Now, when we think about moving forward with foods into clinical trials, there are some things that we need to keep in mind. And as we think about developing the food or the agent, if you will, or the product, we need to think about targeting a specific exposure or dose of something that's based upon science. So you should be able to use epidemiologic studies that talk about servings per week or servings per day to give you some ideas about what kind of a target range you need to be in order to potentially get a beneficial outcome. You need to design the vehicle or the food that is going to assure compliance. Now, everyone in the food industry understands the importance of taste and texture and all these sensory attributes because you're competing in the marketplace with others. But cancer prevention researchers and pharmacologists don't often think about this. But if we want to do clinical trials to show an outcome that is beneficial, we have to have something that people will enjoy consuming. We think about the components or ingredients. What's the right cultivar, the genetics, horticultural conditions, the time of harvest and senescence of the food product, the processing and storage, and whether we're going to be using an extract or concentrate that might put you into the realm of nutraceuticals as opposed to food-based concentrations of bioactives. I can't emphasize enough the importance of analytical chemistry and our folks that are experts in this area to help us define what it is that's in the foods that we're going to be working with. Again, as you develop foods, we move towards the taste, sensory testing, and then, of course, if we're thinking about food-based trials, we have to think about the packaging and stability. And in the modern world, if we want to think about prostate cancer prevention in men with a food product, we have to think about something that's easy to transport, take to work, integrate into meals, travel to La Jolla when you come to a meeting, etc., in order to ensure that folks are getting their doses in the appropriate time and amount. So a key word as you think about developing these foods is consistency. So if we're going to do a food-based prevention trial, we want consistency in the product for every person, day to day, week to week. And I wouldn't do a trial of a food if it was, please go to the grocery store and buy a serving of tomatoes once a day, because we would lose any sense of consistency and we would not be able to conduct a study with sufficient power to get an outcome. The other key point is that your work needs to be reproducible. Whatever you do and report as an outcome in a clinical trial, you want others to be able to reproduce, to carry it further. So another reason to do all this focus upon the product development as you move forward. Now, I'm going to give you 3 examples of food products for cancer prevention that come out of our experience at OSU. There are others that folks are doing in different laboratories around the country, and we'll try to work through this until I run out of time and see if we can't get at least through 2 of these. We'll focus initially on tomato products. We'll talk about a soy bread product, and then we'll focus a little bit on black raspberries and hopefully illustrate some of these points. So, another key issue, food products for cancer prevention and what kind of clinical trials. Phase 1 trials, which we kind of borrow this term from the pharmaceutical industry. I think for many new foods or food products we develop for clinical trials, it's often useful to initially start with a small study in a healthy or a target population. You want to test perhaps the dose, single or multiple doses. Look at feasibility and compliance, meaning the distribution and storage of the food products. So food products are a lot harder to do trials with because of their bulk compared to pills that come in a little bottle. We have to give people crates of food sometimes in the clinic, and The feasibility of just getting it to their car in the parking lot is sometimes an obstacle. Do they have room or place to store it at home, etc.? So these are all the things you learn in these initial trials. We need to test, use different tools to assess intake, monitoring, and various strategies for instilling compliance. These short-term studies are very good at looking at what I put in quotes as pharmacokinetics, or biomarkers of intake or exposure. We can look at this over single-dose intakes or prolonged intake after 3 or 4 half-lives to look at what levels of bioactives may be found in biological tissues. And the wonderful things that we've learned about in these short-term studies is to begin to understand human heterogeneity and variation. It is quite remarkable. In fact, I've been stunned sometimes at the person-to-person variability in some of these outcomes for bioactives when we study this. And of course, in the end, if you're going to be doing clinical trials with a food, you do want to understand the safety issues. We use NCI toxicity criteria and laboratory testing to assess these processes. We then move to phase 2 trials, which are a little longer duration. Often we're targeting our diseased population or at-risk population. The dose is now whittled down to usually 1 or 2. Feasibility, compliance, safety always continues in a phase 2 trial. We're really looking at many biomarkers of exposure, usually in blood, urine, tissue, particularly in pre-surgical studies. And again, this lets us define some of the sources of heterogeneity in the population, whether that be the genetics of the individual, perhaps interactions between food and drugs or other supplements, or how does the dietary pattern of the individual affect how they may metabolize the single food that you're of interest. Biomarkers of activity, then, we're looking at things such as endocrine changes, immune changes, the microbiome, metabolomics, etc. We're looking for changes in the host that may suggest a response to the food that you're providing. And then finally, we're very interested in, if we're looking at cancer prevention, biomarkers of impact in the target tissues and the microenvironment. So we would like to know how is the food, which is altering biomarkers of activity, then impacting the target tissue, particularly looking at biomarkers that are relevant to the so-called hallmarks of cancer. Now, when we move towards phase 3 trials, these are long-term studies, and if we're going to be looking at cancer outcomes, studies such as this are rare and very difficult to do. The cost is enormous, and these limit the number of studies that are going to be done. In all honesty, very few food companies are going to be able to support a long-term cancer outcome study just simply because of the costs. This is something that governments need to do, and unfortunately we live in a world where they're more inclined to support 1,000 therapeutic studies compared to 1 prevention trial. So this is sort of the nature of the game that we're involved in at the current time, but I would love to see more cancer prevention clinical trials underway. Now, one way to help when you're thinking about cancer outcomes is to focus on the high-risk populations. And this could be those who are at genetic or inheritable risk of cancer, whether they're known genetic abnormalities or it's a familial history. These could be in smokers, those that have known carcinogen exposure, those perhaps that are exposed to hepatitis B. We could target those with premalignant lesions or carcinoma in situ of various tissues. And again, we use many biological samples for mechanistic studies, but the ultimate goal in a phase 3 trial is to look at the cancer outcomes. So some of the standards. The randomized double-blinded placebo-controlled trial is an obstacle for food-based interventions. The placebo is often very difficult to create. Patients are aware of what treatment they're on, and you can often see crossover from your controls just based on purchasing products in the stores. There's a lot of background noise, population exposure to the food of interest. If we do a tomato study, I can't eliminate all exposures to tomato products in the community. And likewise, even something like soy, which isn't the most popular food for Americans, is now found its way into many, many different processed food. And we need to think about sufficient duration of exposure, and as you heard a little bit earlier, this dynamic issues about when in the life cycle is it important to have the exposure to impact the cancer. So let's talk about the tomato-based food products for cancer prevention. Here's a label from one of our trials of a functional food because it's tomato juice with soy protein that's been through phase 1 and phase 2 trials. And, we'll talk first about the scientific basis. The original work looking at tomatoes and prostate cancer came from the Health Professionals study published in 1995 by Giovannucci et al. And in their epidemiological work, they found there were no vegetables or fruits that increased risk. Most showed no association, but there was a reduced risk with prostate cancer of tomatoes, tomato sauce, and pizza. And you can imagine what was in the news the next day. Pizza prevents cancer. So, now, you can move to animal models. Now here's a wonderful study that was recently done, and I wanted to show you this one because it illustrates two important points. How to mimic in an animal model something that is derived from an epidemiologic study, but this also shows an overlay of genetics. And this is a wonderful study done with our lab and Dr. Erdman, who's here. And we've got 3 diets in this. We have a control right here. Oops, I'm going to give you the results too soon. The 10% tomato powder diet and a diet that has lycopene, which is the carotenoid that provides the red color to tomatoes. So we now have 2 genotypes here, a prostate cancer model called TRAMP, and we also have a wild type or normal enzyme that cleaves carotenoids. And then we have a mouse model where that enzyme that cleaves carotenoids is knocked out or removed. A single gene out of the 40,000 that we all have. So what did we see and how this impacts lycopene? So first of all, if you don't have tomatoes in the diet, you have none in your blood. If you eat tomatoes, you have quite a bit of lycopene in its different isomers in the blood. And it was very similar whether you got the lycopene from a pure compound or from the tomato. So what happens if we get rid of the gene that cleaves the compound? And you can see that we increase the amount in the blood almost double compared to the wild type. So here's a gene, humans have this, and it clearly impacts how we metabolize carotenoids. So, genetic food interactions, they're going to occur. So what happens when we look at cancer? So most importantly, both tomato and lycopene reduced the amount of cancer. But what was very interesting is when you knock out the gene, the reduction was much less effective in the knockout mice. So here's a good example of how a genetic polymorphism increases the metabolism of a carotenoid and increases a cancer study. Now this work has been supported by a large array of other studies, and I'm just going to highlight one down at the bottom in animals where we completed a study with the Erdman group looking at the interactions of tomato powder and soy germ on prostate cancer and showed that the combination was more effective. So this led us to move forward with some work with our College of Agriculture, our Food Science and Technology program, where we began with those who actually work in plant and tomato genetics and horticulture to get tomato varieties that were rich in phytochemicals. Likewise, worked with our agricultural teams to get the right soy components and extracts. The food scientists literally worked for a year or two to come up with a product that actually tastes good. And indeed, I think at the end it looked right and it tasted like tomato juice without chalk in it. And we very quickly found that as we moved into our phase 1 trials, the compliance was actually excellent. So we completed that trial looking at 0, 1, 2, and 3 cans per day. And I'm going to say at the outset that 3 cans a day was too much, both for compliance, but actually the soy, in all of our soy studies, once we get at a high dose of soy, we get a certain number of people who start to report grade 1 GI distress, and we back off from that because this isn't like chemotherapy where you tolerate high degrees of toxicity. We're talking about prevention, healthy people. You don't want to be bothered by grade 1 GI distress. So the pharmacokinetics and everything else worked out just beautifully. These papers will be published in the early parts of 2017. And I want to show you another slide here. I know this may not be easy to see, but this is showing the heterogeneity of the men in the different groups. And let's just look at the top at the change in plasma lycopene over 4 weeks of intervention. And the 3 groups are 0, 1, and 2 cans per day of the tomato soy juice. And then those who are on no intervention, plasma lycopene decreased, but you can see it's quite variable. And likewise, if you look at the 1 can per day, you see a variety of responses. Getting ahead of myself here. A variety of responses. And when you get to 2 cans per day, you again see a very heterogeneous response, but overall you see a very beautiful mean dose effect. And this was true whether it was looking at lycopene, beta-carotene, or plasma phytoene, another tomato carotenoid. Again, I want to show you how genetics can play into this. Here's a wonderful example where, again, we're looking at the carotenoid cleavage enzyme, a genetic polymorphism in humans that is very common in this enzyme. And look at the estimated prostatic lycopene or prostatic beta-carotene on the left. And there are 3 polymorphisms there shown by the 3 bars at 0, 1, and 2 cans per day. And these clearly had a very strong impact. This polymorphism alone had a very strong impact on the carotenoids and the profiles of the carotenoids that were in the prostate. Now, likewise, on the right, here is a different genetic polymorphism, a single nucleotide variation in the gene having a very different effect on estimated prostatic lycopene. So the genetics and the food and phytochemical effects are going to be very, very important and very intriguing. When we look at an outcome that may be related to impact, here was PSA in this very, very short study, and you can see in the control and the 1 can group, these are men with early prostate cancer prior to surgery. PSA is rising in the controls, and the 1 can per day, it looks like with the 2 can per day group, the PSA, the mean values were actually decreasing. So this is a very, very modest find, but I think very intriguing and stimulates future efforts. Where are we going with the tomato-based food products? We want to enhance phytochemical bioavailability, working with different types of tomato varieties. In other words, the tangerine tomato with polycis lycopene shows 10-fold greater absorption in humans than the red tomato. So we're thinking of moving a little bit more towards that direction. We're looking very closely at the metabolomics of all tomato phytochemicals as opposed to just the carotenoid profiles. We're deeply into looking at how genetics impacts the phytochemical metabolism, and a number of interesting studies are underway looking at novel biomarkers in the preclinical rodent systems. Soy bread. Let's highlight a little of the soy bread. This work was done with Dr. Vodevotes in food science, and she did a great deal of work looking at different mechanisms and different formulations for the soy products. And one of the things we did was add almond to this, which has the beta-glucosidase cleavage enzymes that free the isoflavones from their conjugated form. And she very clearly demonstrated in this work that we could change the pattern of phytochemicals in the food when we did this. Upscaling is always a challenge as you go from the lab bench to the making the bread that you need for clinical trials. So this was a big challenge, getting the right equipment and the right bakers to work with us. In the end, this moved forward with a soy almond bread trial in prostate cancer patients, men with metastatic disease, micrometastatic disease, and rising PSA. A study was done in a randomized crossover where we had soy bread versus soy almond bread. In this, we were able to double blind and collect samples along the way. And 2 publications have come out from this about a year ago, and I would just refer you to both of these in cancer prevention research, one by Jenna and Jarvis, one by Greg Lisinski. The important things that were found in this study is that we observed 4 different metabolic phenotypes in terms of humans using cluster analysis, looking at their metabolomic profile for isoflavone metabolites. Almonds did alter the isoflavone metabolism. Soy alters PSA kinetics, so we found some interesting changes in PSA during the study. But probably most intriguing was we observed that soy impacted biomarkers of anti-cancer immunity. Cancers learn how to put a smoke screen around themselves to hide from your immune system. And the soy seemed to impact the myelosuppressive dendritic cells that are responsible for contributing to this smoke screen in a way that would be more favorable for immune recognition. So this work is being further studied in a trial that will go forward this year in humans. And I'll say a couple words about the black raspberry food products. Again, our food science program, a remarkable effort to create different types of products that could be used in different target groups. And I'm going to talk a little bit about the nectar here. And a clinical trial underway we call the oral carcinogenesis study right now. And this is a complex study, but it's looking at the interactions of gender, smoking, raspberry intake, and the oral microbiome, and how that impacts the mucosal gene expression associated with early cancer. So this is a study where men and women are divided into never smokers and smokers, and will be consuming either the black raspberry nectar or a placebo, which if you didn't really taste both, you might not be able to tell the difference, but we're doing the best we can to create a placebo that works here. And we're going to be looking at these various aspects of the carcinogenic cascade and how these tobacco carcinogens and tobacco exposure impacts the microbiome and ultimately early signs of the carcinogenic cascade in the oral mucosa. So we're looking, this is underway now, halfway accrued, and we look forward to the results over the next year or 2. Now, I mentioned early on something about you got to understand the black box. And so here's an example of some of the lab work that we're doing, and this out of Steve Schwartz's lab with a grad student, Matt Teagarten. And he's using a metabolomic approach to look at the bioactives in black raspberries. And this chromatogram is just illustrative of the over 4,000 compounds that he detected in freeze-dried black raspberry powder using untargeted metabolomics. And so if you're going to think like a reductionist, you're going to be spending many, many decades trying to understand how these 4,000 compounds interact in complex ways to impact the cancer cascade. So get comfortable with the black box. I'm going to highlight one other little finding, this done in the laboratory animal model of black raspberry feeding compared to controls in a mouse, and looking at the murine gut microbiome. And I'm not going to go through everything that's on here, but look at the two graphs in the lower side here. Looking at the different phyla of microbes in the colon, and then the top 10 genera. And you can see a very, very strong effect of the black raspberries in terms of impacting the microbiome. And we think this is going to be very important both in how the black raspberries may impact oral cancer and the microbiome, but I think we are very encouraged to move forward and look at how this impacts the biology of the colon and colon carcinogenesis. When we think about cohorts for food-based interventions, we can think about prevention. That's where a lot of the work that we're doing has been focused. I don't have time today to talk about therapy or survivors, particularly those in remission, but these are very, very important targets for food-based interventions. And in therapy, there's so little we understand about diet and nutrition to impact the efficacy of therapeutics and reduce the toxicity so people can get the right dose on time and have the best shot at curing the cancer. And in survivors, of course, we want to reduce recurrent rates, reduce second primary risk, and reduce long-term complications of therapy. All wonderful targets for food-based interventions. I'd like to thank all these wonderful folks, both at OSU and collaborators at other sites who contributed to the work that I've shown today, and also get a shout out to the Land Grant University System, a government project started years ago that has had enormous benefit and continues to provide great benefit in our area of research. So with that, thank you, and I look forward to questions.
I'm going to actually ask that all four speakers come and sit at the table now, and we'll open up the questions to any of the presenters.